Good evening, everyone. Good evening, and welcome to yet another super amazing session, lecture number 3 on the chapter Anatomy in Flowering Plants. And today we are going to discuss about the types of tissue system. Okay, so this is the lecture number 3 on the chapter Anatomy. We have two more lectures. Tomorrow we will discuss about the Anatomy in Flowering Plants, lecture number 4, where we will discuss about the anatomy of the monocot and dicot root, stem and leaf. And after day we will discuss about the secondary growth, about the phellem, phellogen, and phellodon. So let's get into the session. This is our link of our Telegram group. Those who are new to our channel, don't forget to join our Telegram group at t.me/neat underscore biopoint. Those who are watching the session right now, don't forget to hit the like button and share the video to your friends and subscribe the amazing platform for more updates on biology. So let's get started with the tissue system. What is actually, in the case of animals, we studied about a lot of tissues, names of tissue, and also about the various types of tissue systems. Like we study about the epithelium, epithelial tissue, connective tissue, nervous tissue, muscular tissue, and everything we studied in case of the animals. So just exactly as in the case of animals, in case of plants also you must study about the tissue system in case of the plants. So, in case of the plants, based upon the structure and location, based upon the structure and the location, tissue system in plants can be of three types. What are the three different kinds of tissue system in case of plants? It is the epidermal tissue system, ground tissue system, and third one is the vascular tissue system. What is it? Number one will be the epidermal tissue system. Second one will be the ground or fundamental tissue system. And number three will be the vascular tissue system or the conducting tissue system. In this vascular tissue system only, you will be studying about what all things you will study about there in this vascular tissue system. You will learn about two important components, that is about the xylem and about the phloem. Okay, xylem and phloem. Clear, everybody? Yes. So let's discuss in detail about each of these things in detail. And firstly, we will be discussing about the epidermal tissue system. What is actually epidermal tissue system? Just look at this diagram, everybody. This is a cell. This is a cell. Just imagine. Okay. In addition to this cell, you will see a small layer above it. That is a very first layer. Okay, it will be formed like layers like this. Okay, just imagine if it is formed by layers like this. Okay, so this outermost layer is known as what? Epidermis. So the whole part together you will call them as the epidermal tissue system. Okay, what is it? Epidermal tissue system. It forms the outermost covering of the whole plant body. Okay, so if this is the dissection of a plant tissue, then you will find the outermost layer as the epidermis. It includes the epidermal tissue system, includes the epidermal cells, the stomata, and the epidermal appendages like trichomes and hairs. You may know about the trichomes and hair. I will tell you in detail about that when we come on to the anatomy of stem and root. Okay. What is actually trichome? I'll tell you about that. What is trichomes? Trichomes is actually, I think there is an issue with the mic. Yes, I hope it's fine and perfect right now. Trichomes is actually the hairs, the multicellular hairs, which are actually seen in the stem. Whereas hairs is actually the root hairs seen in the root. Clear? So the root hairs will be unicellular. Unicellular, and the stem, that is the trichomes, will be what? Multicellular. That's very important. So firstly we have discussed about the epidermal tissue system. Epidermal tissue system is forming the outermost covering of the entire plant body. It includes the parts such as epidermal cells, stomata, and epidermal appendages like trichomes, multicellular structure in stem, hairs, that is unicellular structures in the root. Clear? And guys, when you look into detail about this, above this particular epidermis, you will see what a small layer which is referred to as what the cuticle, a waxy layer. What is it? A waxy layer which gives protection. And one more important point you must study, this cuticle is actually absent in roots. Okay, this cuticle is absent in the roots. Done? Yes. So let's first of all discuss about the epidermis. It is the outermost layer of the primary plant body. Point number 1. It is usually single layered. Point number 2. It is made up of elongated and compactly arranged parenchymatous cells. Point number 3. The outside of the epidermis is usually covered by a waxy thick layer called cuticle. Point number 4. And cuticle is absent in the root. Point number 5. So in this slide I have included 5 major points which can come under the epidermis. So what is actually epidermis? Epidermis is actually the outermost layer of the tissue system of a plant body or the entire plant body. It is actually single layer which is made up of elongated and compactly arranged parenchymatous cells. Okay, parenchymatous cells having a large vacuole. Okay, this is a vacuole. So large vacuole. And in case of the stem, you will find, in case of the stem and the leaf, you will find the epidermis layer is covered by a waxy thick layer called cuticle, which helps in the preventing the loss of water, and the cuticle is absent in the roots. These are the 5 major points which you must study in this chapter or in this slide related with the epidermal tissue system. Coming on to the stomata. Okay, coming on to the stomata. Stomata are actually the pores which are present on the epidermis of the leaf. Point number 1. They regulate transpiration and gaseous exchange. Point number 2. A stoma is made up of 2 bean-shaped cells called guard cells. In grasses, the guard cells are actually dumbbell-shaped. This you might know, that is in case of dicot. In case of dicot, it is bean shaped. And in case of the monocot plants such as grasses, you will see it is dumbbell shape. What is it? It is dumbbell shape. Look, this is actually whenever you draw these tomato, you will draw it like this. Right, everybody? Am I audible? Am I audible to everybody? Yes. So I will show you the image of the same. Look, have a closer look at this. That is stomata with bean-shaped guard cells. So the small pore in between, you will call them as stomata. Whereas in dicot plants, you will find a what? Guard-shaped. That is, sorry, bean-shaped guard cell Whereas in case of monocot, you will find a dumbbell shaped guard cell. Clear? Yes. So this is with the bean shaped guard cell. This is with the dumbbell shaped guard cell. Both is this diagram. Both these diagram have been given in the NCRT. So please pay attention. Outer wall of the guard cells away from the stomatal pore are thin and inner wall are thick. Outer wall are thick and inner or towards the stomatal pore are thick. What is the main function of the guard cell? Main function of the guard cell, it possesses the chloroplast and regulate the opening and closing of the stomata. Important. Okay, guard cells actually possess the chloroplast and it regulates the opening and closing of stomata. In addition to this, some epidermal cells surrounding the guard cells will become specialized in shape and size, and they will actually form the subsidiary cells. What they will form? They will form the subsidiary cells. Stomatal aperture, guard cells and subsidiary cells together form the stomatal apparatus. What they will together form? They will form the stomatal apparatus. What all are the components? Stomatal aperture, guard cells and the subsidiary cell. That is, this whole structure you will call them as what? Stomatal aperture. Clear? Just give me thumbs up on the chat box, guys. Is this clear? Okay. Yes. Let's now, this was the same slide that we discussed. That's why we haven't just discussed over here. The outer walls are thin and inner walls are thick. Yes. Perfecto, I hope. Yes, the same slide that we have discussed. Next, coming on to the epidermal appendages, we discussed about the two appendages, that is the trichomes and the root hairs. What is actually root hairs? Root hairs are actually unicellular elongation. Point number and the most important point, unicellular elongation. They help to absorb water and minerals from the soil. Okay. Trichomes are epidermal hairs on the stem. They are usually multicellular. Point number 2. They may be secretory and they prevent water loss due to transpiration. So root hairs is actually seen in case of root hairs may be seen in roots, unicellular elongation, which helps in the absorption of water and minerals. Trichomes are epidermal hairs on the stem, which are multicellular branched, and they also prevent the water loss due to transpiration. Coming on to the ground tissue system or the fundamental tissue system, it consists of all tissues except epidermis and vascular bundle. What do you mean by vascular bundle? I have already told you. Vascular bundle is xylem plus phloem. Xylem and phloem excluding then the epidermal structures. All those structures that comes inside the cell or the tissue of a plant is known as a ground tissue system. They are actually simple tissue. They are made up of parenchyma, collenchyma and sclerenchyma. What are the structures that is coming under that? Parenchyma cells are present in cortex. You must study this: cortex, pericycle, pith and medullary rays. What are those? Cortex, pericycle, pith and medullary rays are present in the primary stem and root. In case of leaf, it will consist of a chloroplast containing cell called mesophyll layer as well, coming under the ground tissue system. So you will be asked the question, I am marking only those, all the tissues except epidermis and vascular bundle. This is the important point. which you must refer at all time. Very, very important. You will be asked to identify the parts. Clear? Yes. Coming on to the vascular tissue system, coming on to the case of a vascular tissue system, it is composed of complex tissues. It is composed of complex tissues, that is xylem and phloem. Clear? This xylem and phloem will together constitute the vascular bundle, that is xylem and phloem. Maybe issue with the connection or something. Yes. So guys, what is actually cambium? What is actually cambium? Cambium is actually a strip of tissue. Okay, it is actually a strip of narrow strip of tissues which actually separates the xylem and the phloem. Okay, this is actually how the vascular bundles are depicted or how they are present. And in certain type of vascular bundles, a narrow strip of tissue like this will be present. This will be referred to as the cambium. And what is this cambium referring to? Cambium is a strip consisting of active dividing cell. I hope now you all can hear me. Just give me thumbs up. Actively dividing cells. So, whenever the term I tell vascular bundles, you must understand that it consists of xylem and phloem. If cambium is present and cambium is absent, it is of two types, open type and closed type vascular bundle. Okay, open type and closed type vascular bundle. I will tell you what do you mean by an open type vascular bundle. Open type vascular bundle means in this cambium is present between the xylem and the phloem. Just remember this, guys. In between this, you will find a cavity, a strip of narrow tissue like this, which consists of actively dividing cell. So, in between the phloem and this is the xylem, you will find this strip of tissue called cambium. Okay, cambium is there, which can form. So, vascular bundles can form secondary and secondary xylem and phloem. So, guys, in morning class we discussed that secondary growth is possible only for a dicot plant, right? In morning class we discussed it in detail that secondary growth is possible only in what dicot plants. And I've already told you if there is secondary growth in a plant only, there will be secondary xylem and secondary phloem will be present. So here it is given that cambium is present. So you will call such type of vascular bundle as open vascular bundle. And what is actually the indirect function of the cambium? It helps in the secondary growth of the plant. So seeing the term open type, you must understand that it is a dicot stem. Those who have not watched the morning session, please do watch it. There I have told you the trick to identify. How can you identify it as a dicot stem or a a dicot stem or a dicot root? I have given you the idea about that in the morning session, how to identify or distinguish between stem you must identify that it is actually what a dicot stem. And coming on to the case, if you see there is no cambium present, they do not form secondary tissue. That is actually the case with the monocot. Okay, and that is actually closed type of vascular bundle. Guys, just imagine in a box, at the opening you're keeping something, so that box will always remain open. So if there is anything present in between, it is open. If there is nothing, the box will be closed. So that is referred to as the open and closed vascular bundle. Open vascular bundle is actually the characteristic property of dicot stem, and closed vascular bundle is actually seen in all monocot plant. These two diagrams are also mentioned in the NCRT. These are also very important: open vascular bundle and closed vascular bundle with cambium and without cambium. Based on the arrangement of xylem and phloem, vascular bundles are again of two types. That is, first one is a radial type, second one is a conjoint type. Look at this diagram, diagram number 1, everybody. Just look at this diagram. Xylem and phloem are arranged in the same diameter. They are arranged in the same diameter. So if the xylem and the phloem are arranged in the same diameter, sorry, 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 it's a mistake from my side. Yes, if they are arranged alternate to each other, if they are arranged alternate to each other or on different radii, look at this, these two are different radius, right? I haven't just checked whether it is radial or something. I just start explaining with the same radius. Actually, this is the diagram where you can find the same radii. Okay, I'll tell you about that. Xylem and phloem, if they are arranged in alternate manner, that is one xylem, one phloem, one xylem, one phloem on different radii, you will call them as a radial type. That is can be seen in all roots, whether it is monocot or dicot root, you will see what radial type of vascular bundle. Whereas if the xylem and phloem are situated at the same radius, you will be seeing those structures in the stem and the leaf. You will have the idea to do all these things or where to apply this when you start solving the questions. Okay. And guys, I have already in the very first lecture of 2022, I have given you the schedule for the entire week. So I hope you might have seen that. And in that, on 7th, on the last day of this chapter, I have included a previous year question MCQ solving. So please do let me know through the comment section whether if we conduct a Menti quiz, a live Menti quiz on that day, instead of just showing the question, I just explaining the answer. If I give you a Menti quiz, will you just come live and do those quizzes? If I get at least 4 or 5 comments, then only we will continue or we'll do that as a Menti quiz, or else it will be a just a session with the help of slides. Okay. Okay, just let me know through the comment section. I will take it out. Okay, yes. So I hope the concepts are made clear. This is a bit difficult one. It's very difficult to understand. You will get confused with entarch, exarch, radial, conjoint, proto, meta, all those things. But we just definitely have to study this. Clear? Clear, everybody? No question is going to be coming out of this from NCRT. Every question from this chapter will be from NCRT. You will get 4 to 5 questions. Done, everybody? So that was all about this short session. Okay, if you have any doubt, just let me know through the Telegram or through the comment section. At least any point you wanted me to explain once again, just tell me through the chat box right now. We will explain those concepts again right now. Just we have 5 more minutes to have the session. So if you just want to ask me something, you can just ask me through the chat box, or else we will end out this class. Is there any doubt? Is there any doubt from any topic that we have discussed Three types: epidermal, ground tissue, and vascular tissue. Epidermal just consists of the epidermal cells, stomata, epidermal appendages like trichomes and hairs. Then we had a look at all of those structures. For monocots, you will see the guard cells with the dumbbell shaped, and for the dicots, you will see the epidermal, that is the guard cells with the bean shaped. Then you studied about the structures that forms the stomatal apparatus, stomatal aperture, guard cells, and the subsidiary cells. Then we learned about what are root hairs, what are trichomes, haploidy like unicellular and multicellular. Then we learned about ground tissue system. Okay, what are the components coming under the ground tissue system also we discussed. Then we discussed about the open type and closed type. If cambium is present, it's open, and if cambium is absent, then it is closed. Then about the radial and conjoint. If they are present in the alternate on different radii, they will be roots, and if they are present on the same radii in stem and leaf, you will call it as a conjoint. So these are just 5 or 6 topics or just points we studied today. That's it. You just have to read out the NCRT thoroughly. I have just completed this session within 30 minutes. So just do study, just read the NCRT. So I think tomorrow is your next test for class 11th. You'll have the test on biological classification, and for class 12th also you'll have the test number 1 tomorrow, reproduction in organisms. So please do attend both the tests in 2022 and 2023. Students do attend the test on biological classification. So Nali, I think there is no further questions, so let's end the class for today. See you. Just comment about the session in the comment section, along with whether you will participate in the Menti quiz on 7th. Menti quiz needed or not? 7th January at the time also, your comfortable time also, let me know. Okay, whether we can conduct it at 10 a.m. or whether it will be at 8:30, you can just let me know. Bye bye guys. See you with the next class in the next day. Bye.